This is our second podcast in our series on the urinary system, and here I'd like to just review some of the cell biology of cells along the uriniferous tubule. So let's start with the renal corpuscle. And as you know, the renal corpuscle consists of Bowman's capsule plus the glomerulus. Now, before we talk about the cells along the tubules, we can define a vascular pole and a urinary pole. And for the sake of simplicity, you might think of the vascular pole as that region of the renal corpuscle where the afferent arteriole enters into the corpuscle and then will ramify into the glomerular capillaries, and that region also where the efferent arteriole would leave the glomerular capillary. So that would be the vascular pole, and almost 180 degrees opposed to the vascular pole, we can define the urinary pole. And the urinary pole is that region in which the urinary space is immediately continuous with the space in the proximal convoluted tubule. We'll also talk about cells of the macula densa in the distal tubule as part of the renal corpuscle. So the first cells I want to take just a couple of seconds to talk about would be the endothelial cells. And the endothelial cells could be the endothelial cells in the afferent and efferent arteriole going into or out of the renal corpuscle, and of course the endothelial cells of the glomerular capillaries. Now the endothelium and the afferent and efferent arteriole are fairly typical endothelial cells. The endothelium in the glomerulus are a little bit more interesting because the glomerular capillaries are fenestrated capillaries, but these capillaries are without diaphragms. This is the only place in the body where there are capillaries without diaphragms. The endothelial cells in the glomerular capillaries contribute to the synthesis of the glomerular filtration barrier. We call that barrier the glomerular basement membrane. Glomerular capillaries have a higher blood pressure than typical capillaries, so it turns out that there's a net positive filtration pressure in the glomerular capillaries of about 15 millimeters of mercury. And you'll look at this in more detail in physiology. I want to highlight the visceral layer of Bowman's capsule. This visceral layer of Bowman's capsule consists of cells called podocytes, and podocytes are modified epithelial cells. The podocytes are analogous to, but they're not the same as pericytes that we talked about earlier in the course when we talked about the blood vascular system. With respect to the podocytes, you'll come to appreciate that podocytes have primary and secondary processes on them, and the podocytes also contribute to the synthesis of the glomerular filtration barrier, the so-called glomerular basement membrane. We can define a parietal layer of Bowman's capsule, and this is a simple squamous epithelium. The location on the parietal layer, as shown in that cartoon, just lines the inner space of Bowman's capsule. The epithelial cells have been shown to have a single solitary cilium on them. We don't really understand what the function of that cilium would be. The parietal and visceral layers of Bowman's capsule, as is the case for all of the cells within the nephron, so all of the cells in this portion of the uriniferous tubule are formed by the same cell type but with different phenotypes being expressed depending upon their location along the nephron. Juxtaglomerular cells are located in the tunica media of the afferent and most likely the efferent arterioles. The juxtaglomerular cells, so-called JG cells, are modified smooth muscle cells. These are the cells that synthesize and secrete renin. And the JG cells and renin may play a role in vascular development. You also know that renin is involved in the renin-angiotensin system. The JG cells may also contain angiotensin 1, angiotensin 2, and angiotensin converting enzyme. And local release of these substances, as well as substances like adenosine, prostaglandins, and various kinins, may cause constriction or dilation of the glomerular vessels. They thus may play a role in tubular glomerular feedback, which then will play a role in regulating glomerular filtration rate. But frankly, we don't understand a lot of the mechanisms about this tubuloglomerular feedback. The JG cells also act in concert with cells of the macula densa and mesangial cells. Mesangial cells are contractile cells, and mesangial cells have receptors for the hormones that we've just mentioned. Angiotensin 1, angiotensin 2, angiotensin converting enzyme, and some of these other hormones. So again, these cells may play some role in tubuloglomerular feedback. Let me talk about the macula densa of the distal tubule. If we look at a nephron, and you can see a proximal convoluted tubule, and it will trace to the proximal tubule to the loop of Henle, come back up to the distal tubule. There is a structure here called the macula densa, shown like this, and the macula densa of the JG apparatus, it's specifically in a part of the wall of the distal tubule. It's at the vascular pole where the distal convoluted tubule begins. So it's at this pole, the notch between the afferent and efferent arterioles. The cells in the macula densa are cuboidal or columnar in nature. We believe the cells of the macula densa monitor urine volume and concentration. It's not fully understood how that monitoring occurs, but we believe the cells in the macula densa also play a role in regulating tubuloglomerular feedback. We also believe that the cells in the macula densa can paracrine signal to stimulate the JG cells to secrete renin and perhaps some of those other hormones that the JG cells might contain. Let me talk about the renal corpuscle and the JG apparatus, and this diagram highlights both the renal corpuscle and the JG apparatus. So we have shown the afferent and efferent arterioles with juxtaglomerular cells, shown like this in green. And remember, the juxtaglomerular cells secrete renin. We show profiles of the glomerular capillaries, so the endothelium of the glomerular capillaries are highlighted in red. We have intraglomerular mesangial cells highlighted by these blue cells within the glomerulus itself, and then there are extraglomerular mesangial cells that are external to the glomerulus, and then the cells of the macula densa, as shown here. There is no basal lamina between the JG cells and the cells of the macula densa, and we believe this plays an important role in the paracrine signaling between these cells. So we can say the JG apparatus regulates blood pressure by the renin-angiotensin system, and the JG apparatus may also regulate glomerular filtration rate. And in that latter case, it may regulate glomerular filtration rate because many of these cells have contractile properties. If we just say a few words about the renin-angiotensin system, and we really should include here the renin-angiotensin-aldosterone system, as you'll learn about in physiology. This system maintains systemic blood pressure, plays a role in tubular glomerular feedback at the JG apparatus to regulate glomerular filtration rate, and What we believe happens is the macula densa cells monitor sodium concentration. The JG cells will secrete renin upon receipt of signals from cells of the macula densa. Renin will convert angiotensinogen to angiotensin 1. Angiotensinogen is primarily secreted by hepatocytes, and then angiotensin converting enzyme, which is found in the endothelial cells of the lung and in the kidney, will convert angiotensin 1 to angiotensin 2. Angiotensin 2 is involved in vasoconstriction of atrial smooth muscle. The posterior pituitary will also secrete ADH, which will cause reabsorption of water, and the adrenal cortex will secrete aldosterone, which will cause a decrease in excretion of sodium chloride from the kidney. This is just an overview. You will go into this in much more detail in your physiology course. Let's talk a little bit about these interglomerular mesangial cells. So they would be these cells shown in blue within the glomerulus. These are within the renal corpuscle. They're between the capillary loops, and they're actually surrounded by the basal lamina of the glomerular capillaries. So they're surrounded by the glomerular basement membrane. These intraglomerular mesangial cells are modified smooth muscle cells. They are directly analogous to pericytes. They play a role in physically supporting the glomerulus. They contain receptors for angiotensin 2, so they may cause a decrease in glomerular blood flow and hence filtration, so they play a role in tubulo-glomerular feedback. They contain receptors for atrial natriuretic peptides, so that will increase glomerular blood flow and filtration, so they'll play a role in tubulo-glomerular feedback that way. But more importantly, the interglomerular mesangial cells are phagocytic cells to clean the glomerular filtration barrier. And this is an important point because at one point people used to assume that the interglomerular mesangial cells were monocyte-derived cells because they were phagocytic. They are absolutely not monocyte-derived cells. These cells are modified smooth muscle cells. Their contractile ability and the receptors they have on them allows them to play a role in tubulo-glomerular feedback, but they also have the specialized ability to be very phagocytic, to clean the glomerular filtration barrier. Now, the extraglomerular mesangial cells are shown here in blue. They lie outside of the glomerulus. They go by several different names, and you may see these names in your reading. So the extraglomerular mesangial cells may be called Pulkeeson's cells or Lacer cells, Gormathti cells, or polar cushion cells. These names are all referring to the extraglomerular mesangial cells. They're mesangial cells. They're considered part of the JG apparatus. They're modified smooth muscle cells. We don't really know what their function is. It has been claimed incorrectly so that these cells secrete erythropoietin. That is absolutely not true. They do not secrete erythropoietin. They may secrete things like interleukin-1 and platelet-derived growth factor, so some people think they may play a response role to glomerular injury, and some people have suggested that they may be stem cells for some of the kidney tubule cells, but that is not totally clear right now. The JG cells and the mesangial cells, both extraglomerular and interglomerular mesangial cells, have been shown to have different types of receptors, like purinergic receptors for ATP and adenosine metabolites, and that also may be why they play a role in regulating glomerular blood flow. That's not something I'm going to expect you to remember, however. Let's talk about the uriniferous tubule. Here we see the glomerulus within Bowman's capsule. We see Bowman's capsule becoming continuous with the proximal convoluted tubule, proximal straight tubule, the descending limb of Henle's loop, the thin limb, the thin ascending limb, the thick ascending limb becoming part of the distal convoluted tubule. Macula densa would be in this area, and then the cortical or arched collecting tubule. That would all be part of the nephron, and then the collecting tubules and ducts, as we have suggested, have a different embryologic origin, and we'll talk about that in a subsequent podcast. Let me say a few words about the peritubular capillary network, or the vasa recta. These terms, peritubular capillary network and vasa recta, are actually defining two different capillary beds. The peritubular capillary network is the network of capillaries that surrounds the short-looped nephrons. The vasa recta is the capillary network that surrounds the long-looped nephrons. The significance in both cases is that the peritubular capillary network of vasa recta originate from the efferent arteriole. Let me say that again. The peritubular capillary network and the vasa recta originate from the efferent arteriole. That is going to have some significant clinical implications for renal pathology, and we'll talk about that later on. The capillary network is part of an atrial portal system, and we'll tell you why that is the case in a few minutes as well. We want to talk about the cell types along the uriniferous tubule. So we'll talk about cell types in the proximal convoluted tubule, in the ascending thin limb of Henle's loop, and the ascending thick segment of Henle's loop, and the distal tubule, distal convoluted tubule, and the collecting duct. Let me go back and talk about portal systems for a minute. On the top you see a typical capillary bed with an arteriole, a capillary, and the capillary going into a venule. On the bottom we can define a venous portal system, something like you may have seen in the GI liver, the hepatic portal system. You have a capillary bed going into a vein, going into another capillary bed, a venous portal system. Here, an atrial portal system, a capillary bed going into an arteriole, going into another capillary bed. This would be the atrial portal system. And so we'll go back to this atrial portal system. Here is the afferent arteriole breaking into the glomerulus and then giving rise to the efferent arteriole, the so-called atrial portal system. Let's talk about cells in the proximal tubule. These cells reabsorb and secrete. Their location in the proximal tubule, they're simple cuboidal cells, but they vary depending on where they're located. We can talk about the proximal or S1 region, which are those cells closest to Bowman's capsule. These cells are most highly specialized for reabsorption. They have an extensive microvillus brush border, as you can see here. They have extensive apical caveoli, so there's a substantial amount of fluid phase pinocytosis going on in these cells. These cells have extensive basal and lateral infoldings with abundant mitochondria. There's extensive interdigitation of the lateral membranes between the cells in the proximal tubule, so the boundaries are not evident in routine histological sections. These cells contain active sodium pumps and cotransporters. They have an active endosomal and lysosomal compartment. These cells secrete dihydroxycholecalciferol to the interstitial space. They also secrete foreign substances and non-filtered substances to the tubular lumen. With respect to the squamous cells of the loop of Henle, there are 4 types. Type 1 in the cortical nephrons, types 2, 3, and 4 in the juxtamedullary nephrons. You don't really have to worry about this. Some people believe there are different structural and functional correlations to the different types of cells in the loop of Henle. The main thing I would like you to keep in mind is that cells in the descending limb are permeable to water, to urea, and to ions, but cells in the ascending limb are not permeable to water. And the type 4 cells, the cells in the ascending limb, are the cells that are not permeable to water. These cells in the ascending limb of Henle contribute to the countercurrent multiplier function that you'll learn about in physiology. Cells in the distal tubule can vary in morphology, and in the distal tubule would be talking about these cells, for example. So they can vary in morphology. They'll have an apical nucleus, as shown here. There'll be a few scant microvilli, as suggested here, and there'll be a solitary cilium. The basal body to one of these cilia is shown right here. We don't really know what the function of that cilia is. These cells in the distal tubule are not permeable to water or to urea. They have fewer features of cells specialized for reabsorption, but they are involved in active transport of sodium and chloride, so you can see extensive basal infoldings of the plasma membrane with a lot of mitochondria interdigitated into those infoldings. So the cells are specialized for active transport. And then, as we have suggested, in the distal convoluted tubule, some of the cells specialize as the macula densa cells. This is just a higher power electron micrograph showing the basal infoldings in these renal tubules. Look at the membrane infoldings and look at the numbers of mitochondria interdigitated in these infoldings. Very highly characteristic of cells involved in active transport. Here is a low-power electron micrograph of the distal convoluted tubule. Note the flat nature of the cells here, but the more cuboidal to columnar shape of the cells in the macula densa. Here is a profile of the afferent and the efferent arteriole, and so it makes the point where the distal convoluted tubule inserts itself between the afferent and efferent arterioles. You see these cells that specialize into the macula densa. Well, let's talk about cells of the collecting duct. We can talk about principal cells. These cells are most common in the medullary and papillary regions of the ducts. So in these regions, these cells secrete potassium, they reabsorb sodium. These cells are impermeable to water unless ADH is present. The principal cells are also known as light cells because of how they stain histologically, and we can contrast the principal cells with intercalated cells. These cells, the intercalated cells, are most common in the cortical collecting ducts. These cells reabsorb potassium. They're involved in active transport of hydrogen ions by virtue of having a hydrogen potassium antiporter and hydrogen ion pumps. These cells are also impermeable to water unless antidiuretic hormone is present. The intercalated cells are also called dark cells, again because of the histological staining characteristics of the cells. Now we can talk about the endothelial cells of the peritubular capillary network and from the vasa recta. As we said, the peritubular capillary network and the vasa recta arise from the efferent arteriole. The endothelial cells in both of these capillaries have diaphragms. The peritubular capillaries originate from the efferent arterioles from the cortical nephrons. These endothelial cells synthesize and secrete erythropoietin. It's also thought that interstitial cells in this capillary bed may also produce erythropoietin, and hepatocytes may also produce some erythropoietin. But remember, erythropoietin is a hormone that stimulates red blood cell production. Again, an important clinical concept here. The cells from the efferent arterioles, the endothelial cells, secrete erythropoietin. The vasa recta originate from the efferent arterioles of the juxtamedullary or so-called long-looped nephrons. The endothelial cells of the vasa recta permeable to water and to salts. These endothelium help to maintain the osmotic gradients of the interstitial space, and they play a role in countercurrent multiplier effects that you'll learn about in physiology. Peritubular capillaries surround the short-looped nephrons. The vasa recta surround the long-looped nephrons. We can see the collecting duct in the diagram here, and then in the hemi-sectioned diagram of the kidney, just want to highlight for you the renal calyces that empty into the renal pelvis and into the ureter. And of course you can see the blood vessels coming into the kidney and the renal vein leaving the kidney. Let me highlight quickly the minor calyx and the major calyces. As you might imagine, the collecting ducts are draining down deep into the medulla of the kidney. They're going to empty into minor calyces. These minor calyces are going to coalesce into the major calyces, and the major calyces themselves will coalesce in the area of the renal pelvis into the ureter. And you may remember from gross anatomy, in the region of the renal pelvis and in the area so-called renal sinus, there's a lot of adipose tissue. Let me also highlight for you here these loops of Henle. Generally, we think of a loop of Henle as the medullary structure, with the loop itself, the turn in the loop, being a medullary structure. And we can define cells in the medullary interstitium as well, connective tissue cells and supporting cells in the medullary interstitium. These medullary interstitial cells are essentially fibroblasts. They're in the connective tissue stroma of the renal medulla. These cells may secrete extracellular matrix components. They may function as stem cells, especially for structures in the papillary regions of the kidney. And some textbooks claim that these medullary interstitial cells synthesize and secrete prostaglandins. And I'm not going to hold you to that, but you may see that in some of your reading. And finally, we can talk about transitional epithelium. Transitional epithelium is also called urinary epithelium. The transitional epithelium lines the renal calyces. It lines the ureters, the bladder, which is not shown on the slide, and the proximal portion of the urethra, which is not shown on the slide. This urinary mucosal epithelium is multi-layered. It has typical dome cells. We talked about this very early in the course, these dome cells. And this is just a histological view, by the way, from the urinary bladder. These dome cells sometimes can be binucleate, and sometimes they can be polyploid. Here are some very big polyploid dome cells. Here's one of these cells that's binucleate.